Okay, happy Monday, everyone. I hope you all had a good weekend, and welcome to the RDM Workshop series, which goes until the middle of December, and we're in session number 4 today. And first, I would like to do the Land Acknowledgement. We pay respect to the Algonquin people, who are the traditional guardians of this land. We acknowledge their long-standing relationship with this territory, which remains unceded. We pay respect to all Indigenous people in this region from all nations across Canada who call Ottawa home. We acknowledge the traditional knowledge keepers, both young and old, and we honor their courageous leaders, past, present, and future. I just want to briefly mention that the registration is open for next week's series and the following weeks. Just as a reminder, we won't be having our session on the 10th next Monday because that's Canadian Thanksgiving, so we shifted it a day forward to the 11th, to Tuesday, and that we'll be having Khalid El Imam, and then on the 17th we'll be having Dr. Aaron Franks, who's going to talk about Indigenous data and open science intersection. If you would like to become a data champion, or would like to know more about becoming a data champion, I would very much encourage you to visit the Ottawa Data Champions website, and we have posted there information about what it is to become a data champion and how to do that. And we also have a brief survey we'd like people to complete if they would like to become a data champion, and we'd love you to join us in becoming data champions. All of that information is available on our Ottawa Data Champions website. Without further ado, it's my real pleasure today to introduce Julia Menon, who's going to talk about preclinical trials and data sharing. So I'm going to stop sharing my screen, Julia, and hand it over to you.
Thank you so much. I will start to share mine, and please confirm that you can see. Can you see this?
Yes. Yes. Absolutely. Amazing. Lovely little bunny.
Yes, a lovely little bunny indeed. Well, I'm very glad to see so many of you today, and I would like to say a big thank you for having me for this session. I hope it will be interesting to all of you. And before we start, I would like to quickly introduce myself, if that works. So very quickly, I'm Julia Menon, as David just told you, and I will be a lecturer today. My current position, I actually have two. I'm the Daily Director for Preclinicaltrials.eu, and this is a platform that focuses on the pre-registration of animal research protocols, and you will learn plenty about this today. I'm also a research fellow for ZonMW. This is a large funding agency in the Netherlands, focuses on biomedical research and innovation. And what I do for them is I do meta-research and qualitative research on open sciences methods, and in particular pre-registration, as you might have understood by now that this is my main topic. From background, I'm a medical biologist, but I actually switched in my career to meta-research, and in particular systematic reviews of animal studies, and I am still highly involved in that field. I'm currently PROSPERO administrator. This is a platform for systematic review protocols. I'm also section editor for laboratory animals for systematic reviews. And another thing I thought you should know about me is that I'm a board member for Young TPI. This is a Dutch initiative that, let's say, pushes the stimulation to the 3 R's in the Netherlands, as well as transition to animal-free research. This is all you should know about me. And before we start, I would like to say I have no particular conflict of interest. And then what can we expect from today? We will have a short introduction regarding some pitfalls in animal research, and then we will move on to pre-registration. What is it and how it can be a solution to these potential pitfalls. And then we'll talk about pre-registration benefits and the platform PreclinicalTrials.eu as an example. If that's okay with you all, I would like to keep questions at the end of the session. We should have about 15 to 20 minutes to discuss this. So please feel free to write them in the chat if you have some burning question going through. Right, so problems in animal research. I know it's a little bit harsh said this way, but I guess the reproducibility crisis is not new to most of you. Since approximately 20 years now, a bit more even, it was shown that most animal studies are not reproducible. And as you can imagine, that has brought a lot of concern regarding both the use of animals, but also the robustness of animal studies altogether. Well, for biomedical purposes, but also for other purposes. This topic is very broad. That could be an entire lecture on its own, but if I had to summarize, let's say, the most recurrent factors behind this reproducibility crisis, I would name these 4, which is first of all concern regarding the validity of animal models. So to actually mimic the human disease or the human, but also lack of proper methods. We'll talk about this in an instant. But so studies that were made in such a way that are not really reliable and trustworthy, but also lack of proper reporting. A study can be amazingly well done, but if it is not well reported, it is going to be quite complicated for someone else to reproduce it, as you can imagine, because you're lacking the details to do so. And last but not least, lack of data accessibility, which will also be one of the main topics today. The data can be amazing, well planned, well performed, well reported, but if it's not accessible to others, this is going to be a major barrier for reproducibility and sharing, as you can imagine. Now, focusing on proper methods and proper reporting, I would like to start with one major issue, which is threat to internal validity. I wasn't sure what your knowledge would be on internal validity, so I decided to take it to the, let's say, easiest level as possible. So here in this example, we have an experimental group and a control group. And as a researcher, what I want to see is the effect of one specific treatment or intervention on a set of results that I have decided to measure. And of course, I want to make sure that my treatment is the only thing impacting my result. However, as any of us can do, we can all introduce some noise or some outcome, external factors that can actually affect these results. Now, normally, this can be somewhat handled if you do blinding and randomization in your study. However, if you don't, you will create some bias, so some systematic error. And this is where your internal validity, so how valid your results are within the study, is going to be threatened. Now, these biases can be introduced at several points in the study. Once again, bias could be an entire lecture on itself, so I'll try to go quickly, but with enough information. So bias, for instance, can be introduced when selecting the animal. If you don't select them randomly, allocate them randomly to the groups, but also during the study itself. This is called performance bias. If you treat the animal from experimental group or control group differently, or for instance, if you're not blinded to the intervention, this is going to have an impact. It can also happen during the analysis of the result or the measure of the result. And this is referred to as detection bias. So as I mentioned, normally this can be reduced or avoided, but if it is not the case, if there's no measure taken against these biases, it will create clear differences between our control group and experimental group. in such a way that the result we want to see are going to be shifted. And we see this here in this example graph. You can see here, where's my mouse here? This is the effect we wanted to see. And so if our initial validity is threatened, it will be either overestimated or underestimated, meaning that the results are not reliable. And of course, that's quite an issue considering the time, the money and the animal spent into such studies. We really want our result to be as true to the effect as possible. And this is something that was actually researched in different meta-research projects. I like this example from MacLeod et al., where basically they looked at different studies and they grouped them based on if they reported randomization or blinding regarding the efficacy of the drug that they had picked. And as you can see here, for any studies who did not randomize, the effect is higher than the one who did randomize, and it's the same is true for blinding. For study who did not report in blinding at the conduct, the effect is overestimated. Same is true here. So I think it really shows that the way we conduct studies have an impact on our result, and that we have to do this carefully and plan well our studies all together. Now, even when the study is really well planned and well performed, some biases can still occur as well during the reporting. I think we all have run into a study once where we thought, I'm not actually sure what they did in that study because reporting was actually rather poor or minimalistic, let's say. But sometimes this goes even further when an entire set of the result, for instance, is completely missing. And we refer to this as selective outcome reporting. This bias occurs when a set of outcome have, for instance, higher chances of being published if they're positive or statistically significant. So it can come from researchers themselves that they think, well, this is not worthwhile to publish this, let's just remove it from my paper. But so in this case, we are missing some outcomes, but it can also come from editor that they will say, let's remove this, this is not relevant in this case. In both case anyhow, these outcomes are missing and it prevents readers and other to have a full overview of what was done. Sometimes this goes even further when an entire manuscript cannot be published. This is what we call publication bias, when there is a higher chance that the study will be published if it is positive, has positive result. Can also be due to the characteristic of the study. If it's a study with a large number of animals, or if it's a new primary study, it might have more chances. Once again, it's quite a shame because it prevents researchers to have a full overview of what was done. And the result of this is that duplication is then is highly likely to happen because other researchers don't know what was done in the past, so they will just duplicate something that was done by someone else beforehand. And as you can imagine, this is research waste of money, people's time, and animals' life once again. Hopefully, pre-registration of animal studies can help with these different pitfalls, and I would like to first define what is pre-registration and then tell you how it can help. So what do I mean by pre-registration? There are several definitions, but I think this one is quite clear. It states that pre-registration is the act of registering research protocols before conducting the experiment. This means that in practice, a researcher will prepare their protocol, and once it's finalized, once they're happy with it, they will upload it or fill up a form in a specific registry or platform which is tailored to this purpose. Usually in their registered protocol, they will state several information about their study, such as the hypotheses they would like to test and the different outcomes they will measure, information about the design of the study, and a clear plan of the analysis they would like to do, so the different comparison and the different tests they want to do. So all of this will be registered. You will receive a timestamp before the start of the experiment. And as you can imagine, this brings quite some transparency. And as we will see now, it also helps to reduce reporting biases because the plan is clearly laid out before the start of the experiment. It is available to others, so there's no, it really helps with consistency in the research. So this type of pre-registration, where someone would upload a document or fill up a form, is what we refer to as simple pre-registration. But there are other types of pre-registration. Maybe some of you have heard about registered reports in the past. So I would like to mention this quickly because that's important. This is the pre-registration I just told you about. You create your study design and then you pre-register your protocol. Then you will go on with your study, perform your study, collect your data, write your manuscript, and then submit it to a journal where you will get through peer review, and hopefully this will get published. Now, registered report is a little bit different, a different format. This is a format that many journals have now, where basically once you're done with your study design, you will submit your protocol to the journal, and your protocol will go through first stage of peer review. And if it is accepted, the journal will keep that protocol. So you'll have a clear timestamp on it, a date that proves when it was accepted. And it will be most of the time hidden by the journal. If your protocol is accepted by the journal, it means that your final paper will be accepted as well, regardless of the data result, regardless of the significance or if it's positive or not. So once this is done, researchers will go on with their study as they would normally do. And then once the manuscript is finished to be written, they will submit it again to the same journal for stage 2 of peer review. And normally after this, everything gets published. So both the protocol and the final manuscript get published together. Now this format, the registered report, is quite new. It's less than 10 years old. It's really increased in the last 2-3 years. Now there's a bit more. It says more than 200, or I think we're close to 300 now, journal accepting registered reports. However, there's not that many, as far as I know, who accept primary animal studies that much, but they do exist. So the difference between, let's say, the simple peer registration and the registered reports, the simple peer registration using a repository or a platform, it is very fast, it's easy most of the time. It can be flexible, so if you have to change your protocol over time, it is possible to do. It will be accessible to all, usually at a date that you decide. However, it does not ensure publication at all, anywhere. It's just a measure of openness and transparency. A registered report, that's a longer process, of course. You have to go for peer review twice. It is a bit less accessible because not that many journals provide this format at the moment. It is stricter, so changes to the protocol won't really be possible, and there will be much more scrutiny actually on if the protocol matches the final manuscript. The protocol as well is only available after publication of the final manuscript. However, the main upside is that you get an in-principle acceptance, meaning that if your protocol is accepted, your final paper will be accepted at the same journal, which is of course very nice. Now, I guess most of you know about pre-registration, but maybe you were not aware of it via clinical work. Clinicaltrial.gov is the largest clinical registry at the moment. started more than 20 years ago, and it really started to take off in 2005 when medical journal editors started to request pre-registration as a requirement. So what I mean by this is that clinical researchers had to pre-register their clinical protocol to have the chance to be published, to be eligible for publication. And since then, the platform has been thriving. We have now several international and national registries all around the world. So this is something that really worked out for the clinic. Now, what about the preclinical, the animal study part? Well, at the moment, there are only two registries really tailored to pre-registration of animal studies. The first one that was ever created is preclinicaltrials.eu, for which I'm Daily Director. We are a Dutch initiative, started in 2018. And a year later, we were followed by a German initiative called the Animal Study Registry. So that was started in 2019. Now, of course, there are other registries that could be used to register your animal study protocol. General registries like the Open Science framework that I guess many of you might know about. This is an American platform started in 2012. However, the difference is that, well, they're quite general, as stated in the name, in such a way that registering with preclinicaltrials.eu or the Animal Study Registry will be much more tailored, much more specific for the animal studies. And now that this is maybe a little bit more clear, what do I mean by pre-registration? I would like to ask one question to all of you, and you can answer in the chat. The question is, have you ever pre-registered a protocol before? It can be any protocol, it doesn't have to be an animal study. And I would like you to state in the chat, A for yes, B for no, but I thought about it, C for no, because I don't know how to do it, and D for no. Oh, I see a lot of A's. This is lovely. Some D's, okay, that's fine. Nice, many A's. Actually, I think this might be the best cohort I ever had. Usually there's no A's at all, or maybe only once. So that's very nice. We'll come back to this in the discussion, but I'm curious to know what type of protocol you have registered. That's interesting. So for the person who said B, no, but I thought about it, I hope I will convince you today that this is a great method to use. And for those who said no, just know the same. I hope you will get to know more about this and also learn how to do it and why it is useful. Oh, registered of systematic reviews. That's right, that's good. Good that you've done that. You should indeed always register your systematic review protocol. I highly recommend it. All right, so what are the benefits of pre-registration? And let's take the example of preclinicaltrials.eu to talk about it. So as I mentioned, a quick introduction about the platform. This is preclinicaltrials.eu. This is how the website looks like, our website. We accept all animal studies, regardless of the type of animals, as long as animals are used. It also accepts all type of studies, exploratory, confirmatory, going from basic research vet studies to translational or preclinical trials altogether. And I know the name may be confusing, but we accept protocol from everywhere in the world, not only the EU. So how do things work within this platform, and what are the benefits of pre-registering? Well, researchers will have to create an account and then fill up a form to give us information about their protocol. That looks, at least the form that we use looks like this. this form really focuses on the experimental design. And it is also compliant with the ARRIVE guideline, the Essential 10. And the reason that it aligns this way is that we hope it really helps researchers to think about their design and their methods beforehand, before the start of the study, but also that it will help them, it will make them gain time on the long run when they have to write up their manuscript. So once the form is filled in, users can submit it to us. We will go through it, and if everything is okay, it will be registered on the platform. So it gets a timestamp, as I mentioned, showing clearly when it was registered, as well as a persistent identifier. Now, the benefit of this is, of course, that it increases transparency. As you can imagine, most protocol information are usually withheld within an institute or an ethics committee, so having it displayed publicly is quite a breakthrough. Also, the fact that there is a clear persistent identifier, like a DOI, actually, is very useful for researchers. It helps them to refer to their protocol in their manuscript or upcoming grant proposal, for instance. It also helps them to be, at least on this side, compliant with FAIR data principle, because this makes their protocol findable and accessible. Another part which is interesting with this pre-registration is that it promotes robust design and better reporting. In the form that we use, we will ask clear questions regarding internal validity. For instance, which method will you use for blinding and for randomization? How did you decide to choose that many animals? What was the thinking behind this? Was there power calculation, sample size calculation, and so on? And so we hope that by asking these questions early, we really help researchers think about the design, guide them in their design, and make it more robust. Now, in the long run, when I forgot I had all of this, my bad. This is what I just said now. I forgot I had this slide. On the long run, once the publication or manuscript is published, so the final publication, it is possible to compare the registered protocol with the final publication for consistency. And as you can imagine, it helps with different biases. First of all, it helps to decrease reporting biases. We talked at the beginning about selective outcome reporting. Well, this is not really likely to happen anymore, considering that researchers have to clearly list all the outcomes that they will measure in the protocol. So it's rather easy to see the consistency of that list compared to the manuscript. It also helps to decrease malpractice. I did not specify this earlier, but I would accept that harking and p-hacking are rather well-known concept by now. If they're not, please let me know, and we can always talk about this again at the end. So harking, creating an hypothesis after the results are known, is also not really longer possible because the hypothesis is clearly stated in the protocol, which is available. And the same is true for p-hacking, considering that the analysis plan is clearly stated in the protocol as well. On top of this, pre-registration helps to increase reliability. Other people, peers, reviewers will usually see the act of pre-registration as good faith from the researchers to be transparent, and it will improve the way people see the research and potentially also see the results. Now, on this platform, preclinicaltrial.eu, it is also possible to link result or data to the registered protocol. And the aim of this is, of course, to increase data sharing altogether, but also to ensure that these data will remain available even if the final manuscript is not published. So this helps in this case to reduce publication bias by making the protocol and the resulting data together available. And we see indeed that pre-registration has effect on publication bias. This is a study by Kaplan et al. Regarding clinical work, but I think it's quite relevant. They looked at a sample of clinical work and the effect of these studies. So as you can see, most of these studies now have these little crosses, which are all beneficial. And then a few of them were null. And then since the year 2000, when registration became mandatory, we see that the overview is much more closer to the null effect than the benefit, meaning that pre-registration actually helps to show the true effect instead of only publishing trials with benefits. The same is true for registered reports. There is a study by SyRCLE et al., for instance, or Allen and Meller, where they looked at registered report and they once again tried to see if the hypothesis in these studies was supported or not. So was the study showing positive data or neutral data or negative data? And as you can see here, for instance, in standard reports, this is a sample just from literature. Most of the studies are positive. They all support the hypothesis they wanted to test. And when we look at registered report, which are, let's say, very true, because in registered report, everything is published regardless of the direction. We see now that the different groups are much more equal. We actually have a lot of study who are not supporting the hypothesis compared to in regular literature. And this is true as well here with estimates from general literature going to 5 to 20, and then registered report showing a much more balanced overview. Last but not least, this platform and pre-registration altogether is also helpful to people who do not register themselves, but only view the platform, only browse the database. So other researchers can actually see what is currently being done or what has been done before. And this can help to avoid duplication of animal studies. But it can also help them to get more information about a specific protocol, better understand how things were done. As you know, sometime in the final publication, it is highly possible that some methodological information just get dismissed. So this platform will actually give them the chance to see things in more depth and hopefully help them with reproducibility. So who benefits from this pre-registration? We have direct group of people who benefit from it. The researchers themselves benefiting from the transparency, the good data management that it provides, their colleagues and peers, because they better understand how things were done. It can help them to prevent these duplication, as well as for reviewers to really have a direct comparison between the protocol and manuscript, also to better understand how things were done all together. Then we have stakeholders who have a semi-direct benefit, journal funders and research institutes. It also pushed them forward to say, well, what I fund or what I publish or the research I support is all well planned through, is all going through well practice, is also a benefit for them, and also a reassurance that what they do is going on the right transparent direction. And if we go even further, we can also find stakeholders with indirect benefit, like the animals. Of course, if this process helps to reduce the number of animals used, that's a benefit for them. And we can see this even further by thinking that it also helps society as a whole by helping transparency. So now that you know a little bit more about the platform altogether, I would like to ask this question, which is, which concern would prevent you from pre-registering on this platform or another? And I would like you to type your answer in the chat. Because a lot of people usually have concerns about pre-registration. And well, first of all, we would like to know more about this concern because we want to find solution on, well, how to make them not concerning anymore. Oh, I see none. That's good. I like that. Is there any other comments that you would like to share? I see no comments. This is a very interesting group. IP, oh, that's a good one. Yeah, IP is something that is quite recurrent in what we hear. Then if there is no further comment, I'll go on. And what I would like to explain now is the recurrent comment we hear as administrator of the platform, and how we came up with solutions to reduce these concerns. So maybe some are yours, or maybe not, but at least you'll learn how the platforms work. So first of all, many people are concerned regarding the money. They say, well, I can't spend money on pre-registration. I'm not even sure that my particular study will match this registry anyhow. They don't want to bother on this. So the good news is the platform, preclinicaltrials.eu, but also the animal study registry, they are free. They're completely free to use, to register, so that's not a problem. And they accept all animal studies using animals. So if you're doing any type of research using animals, your protocol is eligible on this platform. We don't accept, however, clinical trials, systematic review protocols, and so on. Another interesting point for preclinicaltrials.eu is that we accept registration at all stages. What I mean by this is we, of course, encourage people to pre-register, so to use our platform before the start of their experiment. However, we know that it's not always possible for everybody. So we allow registration at different stages. You can use the platform whether your study is ongoing, completed and published, or even interrupted. We accept all stages because we think it's never too late to be transparent, and that it's always useful for yourself and the community to be transparent about the protocol information. Some people also have concern regarding exploratory studies. They think, well, my research is purely exploratory. This platform is not for me. Pre-registration only matches confirmatory studies who are very clear and structured. This is not going to work for me. And well, what we have to say about this is that all studies should be properly planned. And the form that we use is actually matching both exploratory and confirmatory research. So if you have some doubt if this is eligible for you, you can always look at our form and see if it matches your work. But in theory, both exploratory and confirmatory studies are eligible on the platform. One of the other most recurrent concern is lack of time. People think that pre-registration will be a new administrative burden for them. They say, I don't have time for this. This is too much effort. And of course, we hear this, even though we think this is time well invested altogether, considering that it will help you on the long run in the publication process to know what to report and to make sure everything is well reported as well. But to make this easier, we have actually worked on one function that I would like to explain now very quickly. This function is being piloted at the moment in the Netherlands, where we are located. So in the Netherlands, the researchers have to fill in some information for the protocol, about their protocol, for the animal welfare body. And in the Netherlands, most animal welfare body will use one of these software for protocol management. So either PRIS, Ivention, or ticket lamps. So what the researchers had to do is fill in information about their protocol once for the ethics committee, and then they had to do it again for us. And they said, this is too much work. I'm filling up very similar information twice. This is making me waste my time. So what we have created is a direct export, at least for PRIS and Ivention, in such a way that the researchers only have to fill in their information twice. Then they only have to make a few clicks to say export to prerecognical.trials.eu, and then all the information from here get exported right away to our platform. And by using this export, it only takes 1 to 2 minutes to actually register the protocol. So lack of time is no longer a problem. We are also discussing at the moment with ticket lamps to create a similar function. And for some institutes who do not use any protocol software management platform and so on, we also provide tailored documents to help their researchers to register more easily, faster. Another really recurrent concern we hear about is flexibility. Researchers or users will think, well, this is going to kill my scientific creativity. What happened is something changes in my methodology, then my protocol is fixed in that platform. I don't want this. And of course, we have thought this through, and we have made the platform tailored to your research, to the best use for a researcher. And of course, it allows flexibility. So at any time, the protocol can be amended as many times as the user wants. Actually, there's no limit on how many times it can be amended. Just so you know that these amendments will be available on the platform. And last, not least, but one of the main concerns that we have is regarding security and IP. People are really afraid that their protocol and their ideas will get scooped, or that some malicious person will try to go on the platform and steal their private information. And so for this, security is actually our main pole of activity on the platform. We have created an embargo that can be easily requested in the form, which will basically hide the protocol to the public. And then the researchers can decide when to make it publicly available. The embargo is by default 1 year, but it is extendable to a longer date. We accept approximately now 3 years embargo, except if there is a very good reason to extend it longer. On top of this, all registration made on this platform are anonymous, so nobody will know that you personally registered this protocol. The only thing showing up is actually the study center where the study is taking place. And on top of this, in our database, I wish I had more time so I could show this to you, but in the database, only users with an account can access the protocols in detail. For someone who do not have an account, they will see the list of protocols available, but they won't be able to click it. Once you have an account, you can actually click them and see how everything was written up. But so only users have access to this. And to be a user, you need an appropriate affiliation somewhere, in such a way that the platform is really meant to be accessed by researchers and stakeholders in research, like funders, for instance, or companies, but not anybody else. And last concern for today, some people really don't know where to start. They're a bit afraid that pre-registration will be complex. And hopefully we have plenty of guidance for this. The process is quite easy as well. We have different formats that can be used. On the website, we have a frequently asked question tab that is quite detailed. And if this is not sufficient, we have a how-to document that clearly explain with different screenshots how to do things around the platforms, going from how to create an account to how to amend my protocol, for instance. And if this is still not sufficient, you can even book my colleague or I for what we call guidance hours. This is half an hour or a full hour where we will guide you around the platform. We can help you pre-register your protocol live. We can give you a tour, answer any questions. And for PhD students, if there is any today, we can also talk to your PI and tell them more about pre-registration and why we think it would be relevant for your study if this is what you wish. So to sum it up, what do you need to pre-register? Well, first of all, an internet connection, because these platforms, either preclinicaltrials.eu or the Animal Study Registry, are all online. You will need a study protocol. We advise you to have it finished because it will make things easier for you. However, it's also possible to save a draft and come back to it later. You will need an account to the registry and a little bit of your time. But as I mentioned earlier, I think this is time well invested. And if you actually come up with a finalized protocol, it won't take that much of your time altogether. Just some quick information. If you have any question on the topic or the platform, you can always contact us at our info email or my private email. You can follow our updates on social media. We have a LinkedIn profile and a Twitter account. I also put here the website URL as well as the guidance hours. These slides will be provided to you, and all these icons are clickable. So please click them and come talk with us. We'll be happy to help you. And now with this said, I would like to thank you very much for your attention. One last thing, we usually ask people to provide us with some feedback regarding our webinars and workshop. This QR code will take you to our survey. If you have some time, I would highly appreciate that you can fill it in. And then I'm ready to take any question. Thank you.
Thank you so much, Julia, for that great talk. Very instructive. And I think we have one question from Matthew Jeffers. I think he says that my understanding is that during peer review, a reviewer might want to compare the publication to the original study registration for fidelity. If the protocol is embargoed, is there a way to allow only the peer reviewers to have access to the protocol?
Yes, so for now, this has never been requested, but it is possible. So I will be the one making this transition possible, but indeed, we can only provide a sealed version of the protocol to the peer reviewers for them to check it. That's correct.
Okay, thank you so much. Just a question, I guess more generally, is the registry available for people in North America? Let's say someone at our institution wants to put it in your registry. That's feasible?
Yeah, sure, that's feasible. We accept protocol from anywhere in the world. The only thing I would advise is normally we would have one protocol, let's say per experiment. So if you have a large study project with several smaller studies, you should use one form for each of them. That's the only thing, but that's something we can easily, which is actually explained on the website, and that I could always help with.
Thank you so much. Matthew, I see you've got your hand up, so please ask.
Hi, Julia, really nice presentation. I was really curious because you said that part of what you're doing now is to try to make it so you can directly register your protocols like through the sort of animal care committee process, right? So
Yes, that's
those
right.
software that you talked about, are those like private company softwares, or are those some kind of like national system that you have? Like,
So no, so they are private. The institute actually have a subscription to them, and they decide which one to use. We actually kind of highlighted this way how within the Netherlands, which is such a small country, every institute uses something different. Some of them, as I said, had this PRIS or Eventualist, some has Ticket Labs, some only use Word and PDF, and they're happy with this. And that's a challenge for us to, let's say, try to adjust to this. But on the long run, we really want to make this export possible for as many software as possible. And we know a lot of institutes don't use this. And in this case, we provide a guidance on how to copy the protocol, the Word protocol that they have, to our platform. So, instead of scrolling through, I don't know, 10, 15 pages of Word document and thinking, where do I actually paste this? And is that information actually into my protocol that I have for my institute? We create this guidance document that tell you item 1 goes there, item 2 goes there, and so on. So if this is something interesting for your institute, we'll gladly make a guidance document for you. That's always possible.
Yeah, did you like reach out to the private companies with this idea to do this then, or is this something that you're doing through the animal institutes? I guess my question is really, do the animal institutes have their own set of like reasons why they would or wouldn't want to sort of have registration? I think the barriers you talked about are really related to like the individual researcher, right?
Yes, so actually both. So for PRIS, we went directly to the company, but that's a Dutch company. So let's say it makes things a little bit easier, but they were very happy to help us develop this export thing. But a nice example is there is 5 institutes in the Netherlands who use this. And we ask every animal welfare body, please activate the export. And one of them said no. So we don't believe in pre-registration. We are afraid about the IP, and we feel like this is not trustworthy for our researchers, so we will not allow it. And that was very surprising for me, because I told them, you don't have to promote it, you don't have to agree, but just make it possible that if someone wants to use it, they can, and they still refused for now. So I said it's both things. It's both talking with the private companies and talking with the ethic committees. And for Eventualist was a little bit different, because only one institute actually uses it, and their own ethic committee created the whole platform. So then they were in charge of that.
I have one last question, because I think it's a very interesting thing that you raised there about certain committees not wanting to do it. So you mentioned at the start that you're also working with ZonMW, right? And they're a funder. That's right. So is there any, because like, with funding clinical trials, that's kind of a contingency of receiving your funding, right? You're going to do this trial, you have to register it. So is there any kind of push at like the funder level, or to say like, no, this is the rules now, you need to pre-register, otherwise ZonMW is not giving you the money?
Yes,
that would be a way to make people do it, right?
That's right. So we have now 6 or 7 grants for which people have to give them the persistent identifier that they receive once they pre-register. And so the money is actually cut up in different parts, but if they don't give that persistent identifier, they don't receive the next payment. And that's a pilot we're doing with them for 3 years. It will end at the end of 2023. And we're also assessing how the grantees feels about it. What are the challenges they're facing, because they are really eager to push this, but they don't want to make a new burden for the researchers as well. So they're being very careful, but they are ready to make this push. And we also have other health funders in the Netherlands, the Heart Foundation and the Kidney Foundation, now highly recommending. So they don't request, but they highly recommend it in the letters that they send. And we actually hope to spread this to more and more funders in the future.
Yeah, thank you.
I see, do you have the protocol for the intervention you have described, conducting with zone MW? At the moment, this is still, let's say, within closed, how can I say this? This is not officially under embargo, but as it is still ongoing, I cannot provide the protocol of this. That's a qualitative study, actually. But once it is finalized, I would be highly happy to share it with anybody who's interested, of course, in knowing about this, and that should be around the end of 2023. One interesting, one last comment.
Sure.
Our team, so the PreclinicalTrials.eu team and me via Zone MW, we are actually working with other funders on a study with the EVIR Funder Forum. I don't know if any of you have heard about this. That's a forum that gathers different funders within Europe, mostly, I believe. And so right now we are having the surveys among funders to ask them about different open science methods, including pre-registration, and if they would be willing to implement this, or if they are maybe already having different requirements. So we're, let's say, assessing this. And that's something we're also hoping to publish earlier, I would say mid-2023, but I think that will be quite interesting to already know what is being done out there and see the good examples, let's say, that already exist.
Super. Any other questions for Julia? Okay. So I see no other questions, Julia, but I also see that we're approaching the top of the hour, and I obviously want to respect everybody's, though there appear to be now some questions. How is preclinicaltrials.eu funded? From Sally Spendif.
Yes, that's a very good question, Sally. Thank you. We are funded at the moment by the Dutch Ministry of Agriculture, and we hope to renew this funding with them in the future. Otherwise, the rest of our funding is given via grant application for research, as we do research on the topic of pre-registration. So the platform was really started by researchers for researchers, and this is why we also went for public funding.
I think if I could ask one brief question, because I want to respect the hour for everybody. So in the clinical world, we talk about registration, but it seems to me that in the preclinical world that you're mentioning today, you talk about pre-registration. And so what's not clear to me is what is the difference?
I would say that registration is really before the data collection starts altogether, when pre-registration is even before the experiment starts at all, which is quite, I mean, the intervention for me is not even started by then.
So registration, so appropriate registration, prospective registration means that you have not entered the first patient into the study.
Exactly, yes. It's really before anything happens.
So you mean pre-registration is prior to the study design?
So, no, so the study design will be made, but it's before actually allocating the animals to the group and providing intervention. It's before performing the study. But of course, you need to think about your design, otherwise you can't create your protocol. And usually most people will do this after ethical approval, so they know that the protocol is a certain way and won't be moved anymore.
Okay.
Oh yeah, one last point I wanted to mention. On our platform, we check these protocols. We make sure that they're consistent, that there's no errors. For instance, if we ask for animals and you tell me blood sampling, well, that's not that's okay, but it's not the right item to fill it in, let's say.
Sure.
But we don't do an ethical review on these protocols. We don't do a peer review either, so we're not going to change anyone protocol content. The only thing we can ask is for more details, for instance, regarding our form. But that's actually very easy to follow. On the form, you have clear explanation of what to present and then what to write. So we only check this.
Okay. Okay, thank you so much, Julia, for this morning. And thanks to everybody for participating and their questions. Have a great rest of the day. So farewell.